Good day, everyone. Today, our first assigned topic is all about citric acid cycle. But I have a question for you. What comes to your mind when you hear the word citric acid? Because for me, I remember this, the lemon or lime. Because we know that citric acid is can be found in citric fruits like lemon. But I said a while ago that we're going to talk about the citric acid or the Kreb cycle. And I know most of you got confused studying in high school about this. So today I'll explain it step by step. So here's the overview of the citric acid cycle. And I'll explain it later step by step. But let's proceed to the history. In 1953, the Nobel Prize of Physiology or Medicine was awarded to Dr. Hans Adolf Krebs. His discovery of the citric acid cycle gives an impact to metabolism in chemistry. So what's the function of the citric acid cycle in the body or the citric acid? Its function is to help kill harmful bacteria as well as infections on the surface of the skin that can be common in people with diabetes, the elderly people, and the people who smoke. Citric acid can be combined with sodium citrate and potassium citrate to lower the acid levels in the urine to prevent gut attacks. So where can you find the citric acid? The citric acid can be found in the mitochondria. And we know that the mitochondria is produces ATP. It gives energy to the cell. And the citric acid cycle can be named as tricarboxylic acid cycle or the Kreb cycle. So here's an overview of the citric acid cycle. I highlighted the some terms to be easily remembered. So you can read it. So let's proceed to step 1. For step 1 is the formation or the synthesis of citrate. Ako, acetyl coenzyme which carries the 2-carbon degradation product of carbohydrates, fats, and proteins enters the cycle by combining with the 4-carbon keto dicarboxylate species oxaloacetate. This results in the transfer of acetyl group from coenzyme A to oxaloacetate, producing the 6-carbon compound citrate species with the free coenzyme A. So there are two parts: the condensation of acetyl-CoA and the oxaloacetate to form citryl-CoA, a process catalyzed by the enzyme citrate. Second is the hydrolysis of the thioester bond in citryl coenzyme A to product CoA-SH and citrate. Also, it also by the enzyme citrate synthase. Like this. Just like I said, if you're going to notice this one, the oxaloacetate has 4 carbon compound, and the acetyl-CoA has 2 compounds. Because they are combined, with the help of the citrate synthesis, it produces the acetyl-CoA. And that results to 6-carbon compounds. So in step 2 is the formation of isocitrate. The citrate is converted to its less symmetrical isomer citrate in isomerization process that involves dehydration followed by dehydration, both catalyzed by what enzyme? The achotinase. The net result of this reaction is that the OH group from citrate is moved to the different carbon atom. So as you can see, the citrate is not a chiral because it didn't met the 4 different groups. while the isocitrate, you can it is a chiral. And remember that the enzyme acotinase is the enzyme that catalyzes. So let's proceed to step 3. Step 3, oxidation of isocitrate and the formation of carbon dioxide or CO2. The reactants that are a NAD+. What do you call that? The nicotinamide adenine dinucleotide molecule and the isocitrate. The reaction catalyzed by isocitric dehydrogenase. Yeah. So there are three key happenings in this step 3. The following are the hydroxyl group of isocitrate is oxidized to a ketone. Next, the NAD plus is converted and reduced form is NADH. And last is the carboxyl group from the original oxaloacetate is removed as carbon dioxide. It loses, which produce the carbon 5 alpha ketoglutarate. So in step 4 is the oxidation of alpha ketoglutarate and the formation of carbon dioxide. This second redox reaction of the cycle involves one molecule each of NAD+, CoASH, and a ketoglutarate. The catalyst is 3 enzyme called the alpha ketoglutarate dehydrogenase. So this is the enzyme that catalyzes. In step 3, both oxidation and decarboxylation occur. There are 3 products: the carbon dioxide, the NADH, and the 4 carbon species in succinyl coenzyme A, CoA. So there are 4 happenings in step 4. The first is a second NAD plus is converted and to reduce form, the NADH. The second key happening is the second group is removed as carbon dioxide. Then a coenzyme A reacts with the decarboxylation product succinate to produce succinyl CoA. It is a compound with a high energy thioester bond. And that's so in step 4, it is the thioester bond cleavage in succinyl coenzyme A and phosphorylation of GDP. The enzyme in step 5 is the succinyl CoA synthase. Remember, in step 1, the enzyme is they call that synthase. But in step 5, it's the synthetase. The difference between the two is the synthetase is the latter use energy from the breaking of high energy phosphate bond, whereas the former does not require energy. in this step, succinyl CoA is converted to succinyl phosphate. The CoASH is product of this change. The phosphoryl group present in succinyl phosphate is then transferred to GDP. So the product of this change is GTP. When broken, the high-energy thioester bond in succinyl CoA release energy which trapped by the formation of GTP. So anyone can guess what's the function of GTP? It's like the ATP. Because it stores energy in the form of high-energy phosphate bond. In step 6 is the oxidation of succinate. It is a third redox reaction of cycle. The enzyme involved in succinate dehydrogenase and the oxidizing agent is FAD, or we call it flavin adenine dinucleotide, rather than NAD+. Two hydrogen atoms are removed from the succinate to produce fumarate, a 4-carbon species with trans double bond. FAD is reduced to FADH2 in the process. So in the fumarate with trans double bond is an essential metabolic intermediate with both plants and animals, and its isomer with a cis double bond is called malate, and it's toxic and irritating tissue. Succinate dehydrogenase produces only trans isomers of this unsaturated diacid. In step 7, the hydration of fumarate. The enzyme fumarase catalyzes the addition of water to the double bond of fumarate. The enzyme is stereospecific, so only the L isomer of the product malate is produced. So remember that the enzyme is the fumarase, which produce L-malate. Only L-malate is the isomer of the fumarase. With the help of ah with the help of fumarase, rather in fumarate. In step 8, the oxidation of L-malate to regenerate oxaloacetate. So do you have an idea? From step 1, we discussed about the oxaloacetate, and from step 8, there's an oxaloacetate again. It is a fourth reaction of the cycle. It is a molecule of NAD plus reacts with malate, picking up two hydrogen atoms with associated energy to form NADH plus and H plus. The needed enzyme is malate dehydrogenase. So in this step, starting from step 1, go and to step 8, it is called cycle because it repeats itself provided that the additional acetyl-CoA is available in reaction in step 1. So I said a while ago that I'm going to discuss again the whole cycle from the start. And here, first, it starts with the glucose. The glucose has 6 carbon compounds, and via glycolysis, pyruvate is produced. But it will split into 2 just like here, and the pyruvate has 3 carbon compounds. The next step, it will oxidize with the help of pyruvate dehydrogenase enzyme or complex. In this reaction, the CO2 or the carbon dioxide and the NADH is generated. Next step, the acetyl-CoA, which has 2 carbon, combines with this oxaloacetate. You notice I said a while ago 2 plus 4 equals 6. This is the citrate with the help of citrate synthase. Next step, the citrate will be converted to isocitrate with the help of what enzyme? If you say aconitase, that's correct. Next, the isocitrate will be converted to alpha ketoglutarate. If you notice, the NAD is reduced to NADH, and again it loses carbon dioxide. And the enzyme is the isocitrate dehydrogenase. Next is the alpha ketoglutarate. Because it loses carbon dioxide, become 5 carbon compound. Next, before the succinyl, again with the help of the alpha ketoglutarate dehydrogenase enzyme, another CO2 is released and the NAD is reduced to NADH, and then the succinyl is converted. In this step, the GTP is produced, and with the help of the succinyl CoA synthetase. Synthetase. Next is the succinate. The succinate is converted to fumarate with the help of enzyme succinate dehydrogenase. And the QH2 is generated here, which is used to form which is used for production of FADH2. And then the fumarate will be converted to malate with the help of fumarase. Yeah, here. And then the malate is converted to oxaloacetate with the help with this enzyme malate hydrogenase. And again, the NAD is reduced to NADH. And this is why it became a cycle. So what's the result of this cycle? So the result, let me have my pen. We have 3 NADH, that's D, that's D. We have 1 FADH, that's H. We have 1 GTP, and we have 2 carbon dioxide or CO2. Because glucose is split into 2 pyruvate compounds, for each molecule of glucose, the cycle runs twice. So we need to times it by 2. So the result, we have 6 NADH, we have 2 FADH, we have 2 GTP, and 4 CO2. All NADH and FADH here is next to electron transport chain, all known as the ETC, for generation of ATP. So here's the summary of the citric acid cycle. You can read it. So you gain a lot from this topic. Next. And thank you for listening, and I hope you learned something, although it's just a review from high school. I hope that you learn a lot by this topic. Thank you.